podcast. I'm your host, Nick Jikomis, and today I'm speaking with Dr. Rusty Gage. Professor Gage is a professor at the Laboratory of Genetics at the Salk Institute near San Diego, California, and his lab studies how genes and environment interact in order to influence the process of brain development. In particular, his lab has spent many years studying the phenomenon of neurogenesis and adult neurogenesis, the process by which new neurons are born and integrate into the existing circuitry of the adult brain. For a long time, this was thought to be something that didn't happen. For many years, up until probably the mid-20th century or so, people thought that new neurons were born, prenatally in very, very early brain development, and then it stopped. And in adults, you did not get new neurons being born. But over the years, that was shown not to be the case. In fact, neurogenesis can occur well into adulthood and throughout most of the life of an animal. And Professor Gage's lab has been very important for understanding all of the molecular and cellular details of that process. So we talked about adult neurogenesis. How does it happen? What types of things stimulate it to happen? Everything from exercise to intermittent fasting to different aspects of how animals interact with their environment, seeking novel experiences, learning new things. We talked about some of the molecular pathways that influence and trigger neurogenesis in the adult mammalian brain, and all of the things that are involved and related to adult neurogenesis. What are these new neurons doing in the adult brain? How do they work? What happens if this process is disrupted? So if you're interested in the phenomenon of adult neurogenesis, how new neurons can be born throughout the lifespan, this will be a really interesting episode for you. As always, if you enjoy the content I'm producing on the Mind and Matter podcast, please like, share, and subscribe. Don't forget to check out the links in the episode description for how you can support the podcast further, including the Mind and Matter Substack at mindandmatter.substack.com. Hey everyone, I want to take a minute to tell you about a product I use called Everyday Dose. They have created excellent coffee and matcha products with functional mushrooms and other supplements and less caffeine than traditional coffee or matcha products. I actually reached out to them because I've been using their product for about a year or so, and listeners often ask me about my daily and weekly diet habits. They make a really good mushroom-based coffee alternative. It contains micronutrients with antioxidant and anti-inflammatory properties, as well as collagen protein to help support healthier skin, nails, hair, and joints, and the amino acid L-theanine from tea leaves. Each cup has just about 39 milligrams of caffeine. That helps eliminate the caffeine crash that can come if you drink regular coffee, which has much higher caffeine levels. And they use a unique cold extraction process that results in lower acidity than normal coffee. And the caffeine microdose makes it suitable even for someone who doesn't normally drink coffee. This mushroom-based product is made using a double extraction from 100% mushroom fruiting bodies like lion's mane and chaga to maximize the extraction of micronutrients like beta-glucans, triterpenes, and sterols. Other brands don't typically do this, making Everyday Dose one of the highest quality products of its kind. It's gluten, dairy, and nut-free. There's no added sugar. It's paleo and keto-friendly and made with kosher ingredients. There are no grains or fillers, and it is lab-tested to ensure quality. I really like the taste of Everyday Dose compared to black coffee and other mushroom coffees, and they have a mushroom matcha product loaded with functional mushrooms and collagen proteins, so if you like green tea matcha, you'll probably like that product too. If you're interested in a healthy coffee alternative, I highly recommend giving Everyday Dose a try. Check out the link in the episode description or visit everydaydose.com to learn more. If you go there, you can find special offers that they have for getting a free frother and free travel pack of on-the-go doses with your purchase. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it with the first meal of each day, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure. And vitamin D is super important for the proper function of the immune system and for a variety of other things. And there's even evidence indicating that vitamin D deficiency is correlated with more severe cases of COVID-19 in those who get infected. Every time I go into the doctor each year for a checkup, I'm always told that vitamin D deficiency deficiency is very common and I should be supplementing on a daily basis. So visit athleticgreens.com/mindedmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase. And with that, here's my conversation with Dr. Rusty Gage. Does?
Yeah. No, my name is Rusty Gage. I'm at the Salk Institute and in the Laboratory of Genetics. For the last 5 years, I've also been president of the Salk, but that is coming to an end on April 21st of this year. So I'll go back full time to the lab. Work on, I'm a neurobiologist, so I'm interested in generally sort of lifespan of neurons and how they interact with their environment and what influences their development from a stem cell through to a fully functioning neuron. More recently, as a result of the sort of focusing of my attention, I've been concentrating on the general concept of aging. And we use a variety of model systems to study this. 10, 15 years ago, we started working with induced pluripotent cells. But the problem with that is you race the epigenetic profile of the cells, so they go back into an infant state. So trying to study aging with iPS cells is a struggle. So we use a technology where we can directly program fibroblasts into neurons. And when you do that, you retain the epigenetic profile of the person from whom the fibroblast retained. So the fate of the cells has actually changed from a fibroblast to a neuron, but the developmental state of the cell, or its age, its chronological age, is consistent and remains the same. And that's afforded us a lot of opportunities to look at that and focus a lot of attention on Alzheimer's disease, but not regular aging, but also Alzheimer's disease and other neurodegenerative diseases like Parkinson's disease. But aging is sort of a major thrust. We do and most of that's in a monolayer setting. But we do mixed layers of mix and matching cells together, but also use organoids and have a variety of methods that you can drill down on if you wish about how we approach the organoid technology to sort of encourage its generalizability. Mm-hmm. Pause there and let you jump in.
Yeah, why don't we just start with, when we think about aging and cell fate and things at the cellular level for the nervous system, one of the things that's fairly unique about neurons compared to many other cell types in our body is they have to essentially last almost a lifetime, right? So most neurons in the brain don't divide. And so can you speak a little bit about, why that is at a very basic level, and then we'll kind of get into some of the questions around neurogenesis?
Yeah. Well, whys are often hard. You fall back on evolution when you talk about why things are selected for because of their survival value. And so it's not so much a why as a consequence of the environment that you found yourself in. That's a sort of a general statement to avoid the why question. But so what's crucial for neurons, of course, is circuitry and connectivity. And every neuron or many neurons have as many as 1,000 or 5,000 connections to them. And so if neurons were undergoing cell division, this would be a breakage of the connections between them. So one could argue that the nervous system has evolved to a non-dividing state so that circuits could be maintained and memories in particular can be sustained for long periods of time. A good question is, what are the molecular mechanisms surrounding that transition that occurs between a neural progenitor cell, which has the potential for self-renewal and generation of progeny, and a neuron. And we are particularly interested in that transition point. And a sort of very exciting area of research that's emerged out of this recently, or starting to catch on a little bit, is that when cells are undergoing cell division, they're in a state where they use the same sort of energy source that cancer cells use and many other dividing cells. So they use glycolysis. And we showed 8-9 years ago that in iPS cells, when they transition from a dividing neuroprogenitor cell using lots of glycolysis, and I can go into why that would be, they shift into oxidative phosphorylation over a week period of time when they stop dividing. The advantage of oxidative phosphorylation is that you generate a lot more ATP. And for neurons, we think that's sort of the key to their unique function, is that they have so many synapses, have so many activities that they can do in a static state, that they need a lot of ready ATP to do all those things. The advantage of glycolysis, though, is that glycolysis has other shunting pathways where they don't make as much ATP, but they can make nucleotides, amino acids, lipids. So they're involved in generating the bulk that would be necessary for a dividing cell. So that's that molecular distinction and pathways is an area we're spending a lot of time in right now.
I see. So as you go from a stem cell to a mature neuron, in this case, the sort of energetic and metabolic requirements of the cell change when it's dividing. I see. And that's presumably because, when a cell is dividing, it's almost analogous to a baby versus an adult, right? When a baby is growing, it just has different energy and nutritional requirements because it's growing, and then eventually it stops growing and it needs to sort of sit in that state, so to speak, and do what it does.
Yeah, and generate ATP to do lots of things that it wants to do. The other analogy that we use is this energy state of the stem cell or the neuroprogenitor cell is very much like cancer. So a cancer cell is in the same state where it needs lots of bulk to renew itself, to divide, to make more constituents. So we make that analogy quite a bit. I don't know if we're going to go in this direction or not, but I can riff off of that for a bit.
Yeah, before we circle back to that, why don't we just talk about neurogenesis, very broadly speaking. So
Sure.
Could you just define for people what neurogenesis is and how the rate of neurogenesis changes from prenatal to postnatal development to maturity in general?
Yeah, I think you're talking about adult neurogenesis, not in vitro neurogenesis.
Yeah,
right?
yeah, like in an organism.
Right. In an organism, neurogenesis, as I like to encourage people, is not an event. It's a process where cells, and in the central nervous system, there are two locations where this occurs in mammals. One is in the hippocampus and one is in the subventricular zone. There are these niches that are in very specific locations, and they're quite near blood vessels. And they get in the dividing state, they get a lot of nutriment from the vasculature. So we've called this the vascular niche for stem cells. And they're in these niches that exist in other body areas too, as well. So they can either be in a proliferative state where they're making copies of themselves. And as a stem cell, they can either self-renew, that means give rise to a copy of themselves. They're still pluripotent or multipotent at that stage. They can go into a quiescent state or where they are silent for a period of time. And the progeny of a stem cell can also give rise to specific lineages so that you can become a glial progenitor cell or a neural progenitor cell. And these cells then can divide as a committed lineage restricted cell and give rise to themselves. And then from then they generally give rise to one additional cell that will differentiate into a fully mature cell. And then the progeny can either continue to divide as cell or again go quiescent for a period of time. What's crucial about this phase where the cell is lineagely committed to becoming a mature neuron is there's as many interesting things going on while they're maturing as what happens when the cell is fully mature. And so a lot is known about this in the hippocampus, where these cells are in a region of the hippocampus called the dentate gyrus. And they have a period of time during their commitment to becoming a neuron where they're what's called hyperexcitable. They fire action potentials at lower thresholds than mature cells. And this is in part because all the inhibition has not come, all the inhibitory contacts haven't come to the cell yet. So they have this, they're adding a sort of a new dimension or an additional dimension to the cells by virtue of the fact that they respond in a hyperexcitable state. They actually can retain firing for a longer period of time. Again, we think in part because of this lack of inhibition that's coming in. They have generally 4 or 5 different inhibitory neurons that make contact with them ultimately, but that it takes some time for that to come in. So there's even an earlier phase where, this is amazing, where the cells will respond to GABA, which is an inhibitory transmitter, as an excitatory transmitter. So it gets depolarized to GABA. So here you have this cell on its way to becoming a neuron, and it's responding to inhibition by being more excited. And this obviously contributes to their hyperexcitability, because even the GABA that's supposed to be silencing is causing excitability. And then they mature further. They begin responding inhibitorily to GABA, but there's not enough GABA there. So you have this extended period of time where they're hyperexcitable. In rodents, this period is 3 to 4 weeks. So from the time that their last division to their fully mature is about 8 weeks in the rodent brain. But in primate and humans, this period is extended dramatically. So I think the last paper last year in Nature suggested it was more than 6 months, but it had been published in primates that they're out to 6 months or so, but in humans it may be even longer. So that means that this period of time where they're contributing their hyperexcitability to the circuitry is extended. Once they become mature neurons, then they're really not different from the rest of their peers, other than the fact that they have now acquired new information during their maturation that they can contribute to the circuit. So they'll have, there are sort of interesting studies that have shown that if you put an animal into a novel environment, let's say, and then labeled the cells that are born during that period of time with some proliferative dye, and then you take them out, let them mature fully, and then bring them back either to the same environment or a different environment. If you put them in the same environment, then those cells that were born during that time are more likely to respond by expressing an early immediate gene like ARC or FOS than would be other cells. So it's as if they're time-stamped with some information in them about what happened during that critical period of maturation. I don't know if that's too complicated, but it's a fun fact.
Yeah. So a new neuron is born. It starts to integrate into pre-existing circuitry that's already in the brain. But if an animal is in a novel environment while that's happening, the new neurons seem to be specifically related to the novelty there, the newness of that environment.
That's right. That's what the evidence supports. Yeah.
And you mentioned that this is only happening in a couple spots in the brain. Which spots are those again? And is this because do we think that there might be other spots where it's happening, or do they require a very specific kind of nourishment from blood vessels? And there really only are a couple spots in the brain that can really harbor these stem cells.
There are plenty of, I look back on it, there are lots of places that could be sites of neurogenesis. There is evidence that there's neurogenesis in the hypothalamus. There is some evidence that in humans, the second place where it occurs is in the ventricles or lateral ventricles, and they migrate out to the olfactory bulb in lower animals, which have large olfactory bulbs. So there's a contribution, and they go through the same sort of period where they're hyperexcitable there, and they contribute to olfactory memories and that sort of thing. But there is evidence that there's some sort of neurogenesis occurring around the fourth ventricle in the hypothalamus. And then there was this sort of surprising study where there was neurogenesis being reported in the striatum itself. And I think that they're migrating from the ventricle instead of the olfactory bulb, they go to the striatum. But yeah, there is also in the cerebellum, postnatally, up for the first couple of weeks in mice, you see that there's neurogenesis going there, but that pretty much stops in the cerebellum.
But is the main spot for adult neurogenesis, that's the hippocampus?
Yeah, and in a particular region of the hippocampus called the dentate gyrus. So that's the first site. So if you think of a circuitry where the hippocampus is receiving inputs from the cortex, so it's being alerted or brought information in from the cortex, the first place that it goes is to the branches of the dentate granule neurons. And they're just in the inner base of those is a highly vascularized area, and that's where the neural progenitor cells and stem cells of the dentate gyrus occur. And that sort of formation is unique to the nervous system, despite the fact there have been observations of occurring. I think there's actually some evidence for neurogenesis in the piriform cortex, but they never fully mature. I think they get to the state of an immature neuron, but then I guess they die. Mm-hmm. I haven't studied that. I'm just sort of recalling to some published literature.
And so, the sort of biology 101 view of the hippocampus is that it's very important for memory, generally speaking. Do these stem cells that turn into neurons, even into adulthood, are they mostly involved in memory formation and that type of thing, or what are the sort of main functions they tend to take on?
Yeah, the current way of describing this is that they are involved in what's called pattern separation. And what I mean by this, it's a terminology used in computer logic, but you have information coming into the hippocampus, into the dentate gyrus from the cortex. The dentate is very, very densely layered. It's very packed in. And what the role of the young cells in their hyperexcitable state is they send their processes out to the inhibitory neurons in the hilus and activate an inhibition. And because they're so excitable, they inhibit a lot of the cells in the dentate in a non-random way. But at the same time, they're sending out a process that goes to this second relay station of the hippocampus called the CA3. Now, what the data show from many people have shown this is that by ablating or getting rid of these young neurons at critical times, if you present, and this is done by Craig Stark in humans, actually, he's both computational and a human imaging person. And what he has shown is that if you put people in an fMRI machine and then present them with images, okay? So you're flashing a lot of, let's say an automobile, a flower, car, just different objects, okay? Then those are those are the first phase. You're just looking at it. Then he presents to you another object that is either exactly the same as the one that you saw or slightly different. And what happens with age and disease settings that he's looked at is that you lose the ability to make the distinction between closely related objects. So, if somebody says, they bring you this flower, it's the same colors of flowers, but it's sort of twisted in some way, angled in some way. If you have good neurogenesis, you'd be able to see that as the same flower, but it's just oriented in a different way. Whereas, and then you can do these things in the mouse where you they have a touch screen, and you'll show two images and ask whether or not they are in the same location or the same shape. And the closer they are to one another, the more difficult it is for the animal to distinguish between them. But they can make these distinctions between highly similar objects. But then if you delete neurogenesis, you lose that ability to do the finer points. If things are very different from each other, they can see the difference very easily. But it's really working it on this fine distinction. And people have done this not with visual objects, not only with visual objects, but with spatial locations, with emotions. So if emotional things are highly charged or have a big experience or have a big impact, they're easily distinguished from each other. But if you don't have neurogenesis, you have a harder time making the distinction. You know, it's people from the clinical side, people have thought about PTSD, where you have somebody that's had a very bad experience and is associated with some traumatic event, maybe a gunshot or something like that. People that have this PTSD will hear a sound or hear something that sounds like that other one, but they can't distinguish it well. They will react as if they're experiencing again.
So it'd be like if you hear a firework go off and you have traumatic experience from war, you don't distinguish the thud from the fireworks from the battlefield that you used to be
exactly in. And so physicians have used this, and so adult neurogenesis has been linked to depression, and that's where this whole hypothesis about serotonin reuptake inhibitors were being good for that, because serotonin reuptake inhibitors increase neurogenesis over time through a particular serotonin receptor. So that's one practical application of it. But it's also true that it's linked to, say, aging and Alzheimer's disease, when your inability to recognize faces as being folks that you actually know, and it gets progressively worse. You've lost the ability, let's say, to retain if you get distracted, you lose the context in which you were having an interaction previously. So you start over again as if it was a novel event. So it has implications, but the general idea, the way generally people talk about, is this idea of pattern separation, being able to distinguish between closely related objects, emotions, spaces, and time.
Yeah, so you show an animal, a blue car and a red flower. Those are very different. They're going to have very distinct patterns. It's very easy to do that. You don't sort of need new neurons to do that. But when you show them a blue car and then a blue car again, that's just sort of turned in a different way, those two patterns are more similar and harder to separate. And so a lot of these newly born neurons in the adult help that sort of fine scale pattern separation.
That is exactly right. Well said. And so there's a lot of the work right now is on that circuitry, how that can be translated. Most of it's electrophysiology and behavior, but now people are doing single cell sequencing to try to understand the molecular mechanisms that may underlie what are those cells actually doing? What is the event? And it's a very exciting time actually right now because the tools are there to do this. And yeah, good.
So
I'm glad you got that.
yeah. So, I mean, this is fascinating. So, new neurons are coming in to these specific parts of the brain, in particular, the hippocampus. The dentate gyrus of the hippocampus is an important area where this happens. Are these newly born neurons in the adult, are they replacing neurons that have died, or does the brain keep sort of packing in more and more neurons throughout the lifespan?
Yeah, they keep adding new neurons, but on the other hand, it's maybe 1% to 2% of the cells are proliferating at any one time. So as much as we can determine in the rodent, there is a growing. So if you look at an early developing rodent, for example, they have a very thin layer of dentate gyrus. So you can see it expanding over the first 3 months where there's lots and lots of proliferation occurring. That's still building up new memories, building up new experiences about your environment. Then as you mature, you're not going to see as many novel things in your life anymore. So the need for vast amounts of neurogenesis is sort of left. However, what is quite remarkable is that if you expose an animal to multiple complex environments, so you turn them into new environments every day for months, their hippocampal will grow even further. After an adult, you can actually measure the volumetric and the number of neurons growing. So it's a process. And then the other feature of what's manipulative, so it's as if, so normally what would happen if somebody were put an animal in an isolation cage, you have very little neurogenesis, almost stops if you put the animals in a cage with no new experiences. But if you move them into enriched experience, then they start dividing again. And so you can actually manipulate this back and forth by the amount of exposure that they have in their environment. And people are working and have published on the underlying molecular mechanisms that gets that going. And it's circulating peptides are important, as well as neuronal activity. The other big feature that affects neurogenesis is physical exercise. So just movement alone, independently of the environment with it, the movement comes. And from an ethological perspective or evolutionary perspective, you could argue that movement is indicative of coming in contact with new things. So that will stimulate proliferation, and the mechanisms underlying that blood-derived IGF-1 stimulation has been worked out by a variety of people. But also turns out that serotonin input to the hippocampus is also, involved in that activity-dependent increase in neurogenesis. So you have the flip side. You've got lack of activity, restrictions, anesthesia, just no movement, and a lack of visual exposure to novelty are two of the biggest influences on neurogenesis, both positively and negatively. So lack of exercise versus exercise, environmental enrichment versus lack of environmental enrichment. People are doing all kinds of things too, like trying different sensory stimuli. Is it just visual stimulus important, or can you have an impact? One would argue it should be true for olfactory stimuli too, because you're making distinctions between these olfactory. But that might be for animals, that might be going on in the olfactory bulb. But as animals became bipedal, their dependence on olfaction has become less. So I mean, it's almost lethal to ablate olfactory bulbs in rodents. And I mean, think of horses and dogs and how dependent they are on olfaction. We are dependent. It's nice, but it's not an absolute criteria.
Yeah, so the amount of neurogenesis in different parts of the brain probably varies from animal to animal depending on its, its
It's
needs.
biology.
Yeah.
It's biology. Yeah.
And so it sounds like, you said, so in terms of thinking about like the things that trigger neurogenesis, it sounds like some of it comes from just patterns of activity in the brain itself, the way that neurons are firing in the hippocampus and elsewhere. Some of it comes from things that are circulating in the bloodstream, and those things change as a function of, novel environments and movement and exercise and things. What are some examples of the sort of key molecular triggers that promote the differentiation of these stem cells into mature neurons?
Yeah, no, actually, it goes back to my original discussion that we were having. So we do know. I'll take it this way. We do know, for example, that this serotonin activation from the serotonin is the serotonin neurons of the RAF-A fire when the animals are in an active setting. That those synapses onto the neuroprogenitor cells, and they activate through serotonin receptor the activation of genes involved in proliferation, CDKs and things of that nature. So there is a biological part of it. There's also, as I said, IGF-1 can get through the blood-brain barrier to stimulate neuroprogenitor cells that are near the blood-brain vessels. It's interesting. Their location is key to a large extent because they're getting access to the vasculature during those times. And what is also happening is that there's an increase in angiogenesis. So there's actually increasing of blood vessels, small auxiliary vessels are branching out in these cases where you have a proliferation, or when you have running experiences going on, increase. There isn't as much, I think, on the environmental enrichment as I'm trying to recall whether or not people have shown increase in vascularization with enrichment. I guess it depends on the size, the enrichment, and how much movement. I think it's really a movement-dependent phenomenon there, I think.
So
it has a vascular component, angiogenic component, direct stimulation from increases in cytokines and growth factors in the blood that are released as a function of movement. But there's also central mediated pathways like serotonin that mediate it. So it's not a single event.
And so exercise and movement can be a stimulus that promotes neurogenesis. And if there's also stuff circulating through the blood, what about just dietary components? If you just change the composition of an animal's diet, does that have an effect on neurogenesis independent of movement and other things?
Yeah, there's actually been a fair amount of work on that. The trouble a little bit with that is that mechanistically it's less well known what's causing that. But one thing that has been repeated is this dietary fasting, intermittent fasting also increases neurogenesis. I'm not sure what that is about, but it might play a role in this discussion we had earlier about metabolic shifts. So from glycolysis, glycolytic activity, and dependency on glucose for generating lactate during the glycolytic pathway versus sort of generating pyruvate so it can go into the TCA cycle. So these metabolic elements can be affected by diet and might influence whether or not it shifts. And that's a speculation on my part, but what I'm interested in following up on.
Yeah, no, the metabolic side of this is interesting. It makes sense that as a cell is, changing what it is, going from one phase of its life cycle to another, it's naturally going to have different kinds of energetic requirements. And so there's going to have to be metabolic shifts there. So you mentioned when the cells are proliferating, they sort of use energy in the way that cancer cells do. And after all, cancer cells are really just, very fast dividing cells. And so that piece makes sense. It sounds, when people talk about neurogenesis, we tend to talk about, helping with pattern separation, memory, and these things. Is it always a good thing, or are there times when neurogenesis can be a bad thing? Can some of those proliferating cells become cancer cells?
There's not, we looked at that for a while here, seeing whether we could do that, generate by overexpressing cancer genes in the cells. And you can force them to do it. But spontaneously, there's not a lot of evidence for that. And even the gliomas that people study, I'm not convinced that the evidence supports the fact that they're derived from the dentate area. It's more likely derived from a proliferating glial cell that then has the capacity to turn, rather than really a committed neural progenitor cell. But it is very interesting that there are no neural cancers. So once a cell's committed to a neuron, you don't see neuromas coming from that. They're always coming from, if anything, very early development where the neurons are still in a proliferative state. So it is conceivable, but there's not a lot in my read of the literature. There's not a lot of evidence supporting the fact that that's a site where neuroblastomas are coming from.
And so when the cells are sort of coming out of the niche where the stem cells can be nourished, they have to differentiate into neurons or other cell types, and then they have to physically get somewhere, right? Like how are they physically moving from one spot to another, and how far do they actually travel? Is it fairly local, or can they travel long distances?
It's pretty local. So think of it. So you had a blood vessel. I'm trying to give you an analogy here, and the stem cells would be sitting right in the cuvette there, getting all their nutriments, maintaining, and also their oxygen levels are dependent upon their proximity to blood vessels. As they move away, they are less proliferative. So that's when their proliferative niche really occurs, with these nutriments and their oxygen content. When they move away, so they can, there's sort of morphological evidence suggesting that along these layers, you'll have glial cells on the base, and then you have neurons that are focusing up into what's called the molecular layer, where the dendrites all receive the inputs from the cortex. And these cells can migrate along the surface of some of these cells, but the distance that they're traveling is really not very far. It's a little lateral from the vasculature, and they move up, 15 microns to 30 microns up into the granular layer. What happens, interestingly, during development, when there's such a thin layer of granular neurons initially, is that they're born in the base, and the ones they're born earliest, they move up, and the other ones just move into the place below them. So they're not migrating up to the top; they're coming up from the bottom, and that's been demonstrated in a variety of different ways to show that. So they're not really moving very far, and they're laced all along the inner, called the intergranular zone, which is where they're born, but the total distance traveled is not much. Although they're dendrites, of course, and when they're maturing, their processes have to extend out over long distances. But that's and the directionality of those dendrites, if you will, is really very important. Mm-hmm. The molecular mechanisms controlling that is well studied, and there's Wnt signaling, which is really important for the non-canonical Wnt signaling. So the canonical Wnt signaling, beta-catenin, et cetera, is involved in the proliferative state, but then it shifts over to the non-canonical, and that controls the directionality of the dendrites as they migrate up into the molecular layer.
I see. So you've got this population of stem cells that live near blood vessels, and they're sort of essentially standing in line, waiting to get right up next to the blood vessel. And the ones that are right there get all of the signals circulating in the blood that might tell them to start dividing and becoming a neuron. And then the cell body of the neuron does physically move, but not an incredible distance. But once it gets to where it's going, the branches of the neuron do grow outwards and upwards and in different directions to make all of the connections that it needs to make.
Yeah, just one refinement is that when they're next to the blood vessels, they are in either a quiescent or a proliferative state. They're not becoming neurons. They have to move away from the blood vessel.
I see.
And they shift from using Wnt signaling, Wnt3A, and those kinds of proliferative signals into the non-canonical form of Wnt, where you're Wnt7 and Wnt5, which uses and it stops the proliferative kinds of signaling at that point.
I see. So they really are distinct phases.
They're different.
You proliferate, and then you turn into
and you
something.
move away from the vasculature where you're getting all these proliferation signals.
I see.
You move into the parenchyma where you're involved more in this differentiation process. And that's where this metabolic shift is happening as well.
Interesting. And so so we've mentioned that the things that can stimulate neurogenesis, just novelty in general, movement, probably different aspects of diet, just, being exposed to new sensory information that you haven't seen before, learning new skills. And it sounds like there can be a significant amount of neurogenesis in the sense that you could see it macroscopically. So you mentioned that you can just see that, the hippocampus can grow. So would this be like, I know there's a famous example of like the London taxi cab drivers. They have to memorize every road. And if you image their brains as they do that, their hippocampus literally is physically bigger in the imaging pictures that you see. And so there's enough neurogenesis that it can lead to that level of macroscopic change.
Well, the concern about that study, as true as it is that are people that are good at memorizing things and already have a big hippocampus, are they more likely to pass the test for taxi drivers or even choose that application? You need to do the study where you have a measure their hippocampus prior to even thinking about wanting to become a taxi driver, and then all that experience leading up to it. I'm not saying it's not causing that. It's just that particular experiment doesn't, count out the possibility that there was a selecting for that kind of behavior because you had that capacity.
But it sounds like neurogenesis is happening. It's not necessarily an ultra rare event, depending on the type of experience an animal is having. So, like, just to give us
I'll give you one example.
Yeah.
So a postdoc in my lab called Henriette von Prague was the one that discovered that running could increase neurogenesis. And what you do is you inject the animal with a false nucleotide called bromodioxyuridine, and it's incorporated into the DNA of cells that are undergoing cell division. And you can stain it, it turns black. And so she injected the animals and ran them in a running wheel, and then looked at their brains later, and she didn't have to look under the microscope. You could see the black lining inside it. And she was showing it to everybody in the lab that it was, and it was, you can quadruple the number of dividing cells by some of the experiences or by the running in running wheel. Just running, it wasn't doing anything extraordinary, just allowing them to run. It wasn't forcing them to run. It was just allowing them to run. So that gets to your point that it's not the whole hippocampus, it's not the whole dentate, but it's adequate enough to actually see it visually.
And does this ever go away? So if you took elderly animals, does neurogenesis ever turn off?
Absolutely. And it can turn off experimentally. So in your life, if you're under severe stress, it definitely shuts down neurogenesis. So there's a cortisol link there to the proliferative event. If you do physical restraint, if you put animals into a physical restraint so they can't move around at all for a period of time, that'll dampen neurogenesis dramatically. So there are ways you can do this by doing the opposite of increasing exploration or increasing activity. You can do it that way. Some people have claimed that even depression, anxiety, those kinds of things, but that's also linked to the lack of movement oftentimes, where people are not moving around, they're just sedentary, not eating right. And so it's harder to link activity of neurogenesis to a cognitive state than it is to the primary readout, which would be movement or exploration. Now, to your point, in rodents, it clearly decreases with age to the point where, so rodents have let's say mice have a lifespan roughly in captivity, the way we breed them, about 2 years, 24 months. And neurogenesis peak would be around 6 months, maybe a little bit younger. And by 9 months, you're definitely declining. And by, 18 months is very, very little going on. However,
what would that correspond to in a human? Is 6 months sort of like an adolescent and 18 is like a middle-aged person?
It's so hard to make that extrapolation, but yes.
But at 6 months,
think of 2 months as a teenager. Think of 4 to 5 months as a mature adult, and 9 months. So this is the real trick, and that is that in the wild, the half-life of a mouse is 9 months.
Yeah.
So what we're doing, we're keeping these animals alive for a much longer time.
So 6 months is a fully mature young adult mouse.
Oh, absolutely. Yeah. Absolutely. I mean, for some people, yes, that's fully mature, but then it drops dramatically over time. However, even at 12 months, 15 months, you can take a mouse out of their isolated cage, put them into a running wheel, and increase neurogenesis again. So there remain stem cells. Many of them are quiescent at this point. And partly because they're living in these little cages, not experiencing a whole lot of new things. And the need for neurogenesis, is not, and this is where the interpretation of that would be just like when older people, they're more sedentary, they may be bedridden, they're not traveling and not seeing a lot of new things. The question that faces us is there a real biological clock where it decreases, like many things, or is this just a function of activity? And you can maintain high levels of neurogenesis if you kept experiencing new things, kept physically active, and so that that it's two ways of thinking about it, but experimentally testable.
And you mentioned before the importance of serotonin as a signal involved in the process of neurogenesis generally. So when we think about serotonin, what are some of the drugs that tend to promote neurogenesis? You mentioned SSRIs. Do other serotonergic drugs do this? Do other completely different types of drugs promote neurogenesis? So just so to clarify, when we say neurogenesis, you need to break it up into its process. So there's some drugs that have an impact on proliferation. So you're increasing the pool of cells. Then there are drugs or mechanisms that drive the cell from a proliferative state into a differentiation state. So that's how it's best to think about it, rather than saying just a drug affects neurogenesis. It really, is it affecting the proliferative state, or is it affecting the differentiation state? And so there are, like I said, serotonin receptors on the neural progenitor cells that's involved in their proliferative state. And, there are a variety of drugs that can be used to induce differentiation. The concern that you have there is, are you taking them out of cell cycle and driving the pool of dividing cells all into a differentiated state, and you lose the pool that you're drawing from under a more natural setting? So when thinking about using pharmacology to induce or potentiate, quote, neurogenesis, I like to encourage people to think about, what is it that you really want to do? Do you want to increase the pool? Do you want to increase the available cells that can go into the circuit, that can contribute to an ongoing event over the next few weeks? Or are you trying to retain the pool for later in life? Or actually the third piece, we think even more so in humans and
primates is that there are a fair number of quiescent cells. And so the question is, how do we reactivate those quiescent cells to bring them back into cycle? Or, and I'm sorry to make this even more complicated, but it's not just differentiating the cells, but you want to make sure that the cells differentiate at the right pace? Because there is this concept of neoteny where the maturation of an organ, of an individual, of anything is timed so that you can have the appropriate amount of experience during that maturation period. Think of it in terms of, your kids when they're born. You want to make sure that they have that adolescence and that those childhood experiences. You want to rush them all the way through to become mature adults with all the responsibilities of a mature adult when they're 11 years old. You want to make sure that they have that opportunity to mature. And it's the same with cells, and certainly same with neurons. You want to make sure that they have that full ability to make all the appropriate synapses, to let their dendrites get as long as they can before they become contributing members to the network.
So you wouldn't necessarily want to pharmacologically induce proliferation or differentiation if it wasn't also in the right context, meaning in a novel environment or in the context of learning something new.
I think this is supposition, but it's based on the accumulated data and the knowledge that would be my approach, is to think of it now in an aging state. I think it makes a lot of sense to pull cells out of quiescence, to activate the quiescent cells into a proliferating state. And then in the context of experience, you would have them naturally mature because you've increased the pool. Then by giving them more experiences, they're being integrated by virtue of the experiences that they have. So it to me, it likely strikes me that in cases where neurogenesis is stopped due to disease, aging, anxiety, whatever the onset is, getting the quiescent pool back in cycle and then getting the individual to have enriched experiences makes a lot of sense. I remember talking to a clinician who dealt with PTSD and depression, and their view of it was that they felt that the SSRIs work best if you can link them to physical exercise. So the whole clinic was making sure. And so one of the interesting things about that, and again, I'm not a clinician, but just from and there are books and papers written about this, so I'm not making this up from whole cloth, is that one type of therapy after the therapy is going well is in PTSD, if you re-expose them in a safe environment to that stimulus that was evoking a traumatic-like event, and you expose them, you give that stimulus and they react, but it's now in a very safe environment. You do it multiple times, they'll begin to dissociate that stimulus from the initial event and link it more to the new events that are where it's happening. Does that make sense?
Yes. It
made sense to me.
Yeah. So it's allowing someone to allowing that traumatic association to disintegrate and effectively be replaced by a new associating
with a new experience, which is non-threatening.
Yeah. Interesting.
Non-harmful.
What are some of are there particular serotonin receptors that are important with respect to?
There are. It's 5-HT. I think it's 1A. It may be 1B, but I would have to look it up. But there are very specific receptors that people have been targeting.
Yeah, and I know that there's been some discussion. There's been quite a lot of research excitement going on with respect to psilocybin and other tryptamine psychedelics. And I believe there's some evidence out there that they promote neurogenesis in animals, but I'm not quite sure. Is that true?
I'm not enough of an expert in that. My understanding and is that most of the research that's been done at a sort of morphological level shows that there's increase in plasticity of the synapse, so they can somehow open a plastic window. I'm not sure how mechanistically that has been really evaluated. I'll bet you somebody has done, psychedelics in a rodent with VRD labeling to see whether or not it increases neurogenesis. I just embarrassed to say I don't know the literature on that.
Oh, okay, yeah, no worries. What would you say some of the latest discoveries and surprises are in the field of adult neurogenesis, broadly speaking? What are some of the things that we've learned recently that we didn't know, I don't know, 5, 10 years ago?
Yeah, I think one of the I think people speculated that this was true, but a group in Spain they have this amazing fast autopsy situation where they can get brains from people at all different ages and different disease states. And they were the first to really show that there was a decrease in neurogenesis and Alzheimer's disease that is much greater than decrease of natural aging. And then that was replicated by two other groups, one in Chicago and another one at Columbia. So I think we had the idea was involved in aging, even in rodents, but that it specifically elevated or decreased more in AD. And they've gone on to have a link to microglia. So that I think is there's one also, I think, an interesting set of experience that we didn't know. I don't know how long this is published. There's a group in Canada that believes that increase in reports strong evidence that increases in neurogenesis, like with running or something like that, can increase the rate of forgetting. And so, and they talk about the importance of forgetting. You don't want to remember everything. But the other way of saying that, and what other people interpret it mean, is that it's not so much they're forgetting it, is they are generalizing about one event is similar to, but exactly not the same. So it gives you a broader focus or a broader range of things to categorize them rather than just a very specific memory. I mean, I think for it to intuit that a little bit, think of, you have, here's a good example. I don't know if it's a good example. This is my best one I can come up with at the moment. So this is a clear plastic spoon, right?
Mm-hmm.
What's this?
That is looks like a metal spoon.
Okay. If you didn't have neurogenesis, one would argue that you would say they're different. But really, the truth of the matter is they're also quite similar.
Mm-hmm.
Functionally, they're quite similar. And one way of interpreting the importance of neurogenesis is not only can distinguish between things, but also helps to determine the relationship between things. Mm-hmm. And that is, this pattern separation is followed by what's called pattern completion. And that occurs, we believe, in the CA3, the next layer. So they're separate, but they have a relationship between them. They're linked in some way.
I was going to say, it reminds me of what you were saying earlier about pattern separation. So on the one hand, in order to form a general concept, say, of spoons and what spoons do, you have to be able to identify slightly different individual cases of spoons. And if you can do that well, which in neurogenesis you said was important for, that then should also aid in the forming of just the concept or the category of spoons.
That's right.
What are some of the big unanswered questions? What are some of the things that your lab is studying
I hadn't
today?
even talked about. So another one, I'll tell you another new observation, I think, in the human field is that that embryonic stem cells in the brain have a signature that's very much like the adult cells. So human.
Mm-hmm.
But those signatures are very different in other species. So there's something unique about that. Then it turns out that, and this is, I mentioned this to you before, this concept of neoteny, where, it's important to go through a time period to get lots of experience before you mature. Well, big area of investigation right now is the fact that it differs dramatically in different species. And humans, it's much, much longer. So it takes much longer and this is something I've worked on in many neural systems, both in vitro and in vivo, is human cells take a very long time to differentiate into fully functional cells.
Like weeks, months?
Months. Yeah. And versus weeks in rodents. And, you can go ahead and ask your question about why. Why is that important? But I would only speculate that period of time when they are maturing, they are playing a special role in the function of the dentate gyrus, because they're adding this hyperexcitable state in there. They're doing other things. And we're particularly interested in understanding what information are they actually contributing. I'll tell you this. We just have a paper coming out where we went into it thinking, well, these young cells, and they're hyperexcitable, maybe they're if you looked at them transcriptionally at the single cells, you're measuring single cell transcriptome for all the young cells versus the mature cells, you might guess that these young cells were expressing more genes and a unique set of genes because they're contributing something more. But it turns out what we showed was that they're transcriptionally less active than mature cells, which is paradoxical in my mind. But then they're also pretty young, so they haven't developed a full repertoire. So that transcriptional profile is reflecting something different from the mature cells, but it's given us a hook. Say they are different, and now we're into this now, trying to find out what is these pathways that are important for understanding the cells, and the paradigm is that you take an animal out of a home cage, individually housed home cage, put them into a very rich environment where they're seeing all sorts of things for the first time, and then you shortly take single cells from the hippocampus and you sequence all the cells, but you first sort them for the cells that responded by expressing a gene called FOS, the immediate early gene, or others that you can use. We sort those cells out and compare them to the cells in the mature dentate that responded to the same symptoms, the same animal. And that's where you're seeing the cells did respond, but the impact of the environment on the young cells is much less, despite the fact that they're excitable, whereas the mature cells have a lot more genes being expressed. Interestingly, and this is a broader issue, is if you look in the CA1, CA3, which are other areas in the hippocampus, their expression level is really low. They're not making much of this. A lot of the information, the biggest impact is on these granule neurons. Even though only a few of them are firing, the ones that are firing are responding with massive numbers of genes. So we're very excited to try to dig into this, understand what that molecular information is that is going on. I'm not sure how we got down that road, but
yeah, no, I was just asking about exciting new stuff that's going on. I want to ask a more general question. So the dentate gyrus of the hippocampus, this very important region where a lot of these new neurons go, what happens if that region is just damaged, generally speaking, to an animal or to a human? What kinds of problems do they tend to have?
Yeah, and actually, prior to the involvement of neurogenesis, so in behavior, which really started happening in the 80s, 90s, but in the 70s and earlier, people would do selective damage to the dentate gyrus, and you do see, profound, profound differences. I'm trying to think of a test that was used in those days. I think it was there were a lot of maze type of tests. Let's just say that they clearly have selective damage. It's hard though, because this is a very complex structure. Think of the hippocampus is in your brain. It comes like this and it goes up here. It's about the size of my finger. And in the middle of it is a band of cells that form a U that go all the way through like this. So trying to kill or destroy that curved C-shaped structure as it winds its way through there selectively has always been sort of
you can't cleanly and specifically hurt that region. It's
very hard to cleanly do that. And the other part is that the rostral and the caudal part of the structure are doing different things. The current thinking is that I mentioned to you that there was both an affective and a cognitive component, or emotional component, and sort of learning memory. And the more rostral portion is thought to be involved mostly in the cognitive components, and the ventral, which is closer to the amygdala, is thought to be more involved in the affective components. So it's different even along its route, even though it's playing the same role of pattern separation. One is thought to be more than the other.
But I would imagine in general, if you get some damage somewhere along that structure, they're going to have some kind of learning and memory or navigation.
Oh yeah, no, this is. I'm sorry. Yeah, this is the classic case of hippocampal loss. This is where people think of. There was a very famous case in Canada called HM. Individual that had damage to one temporal lobe in hippocampus, and then had seizures and removed the other hippocampus. And the person had a very difficult time acquiring new information. It was sort of a very classic text. That's what the textbook description is of hippocampal damage, is this inability to integrate new information into you have some past memories that you can recall, but acquiring new memories is particularly disadvantaged in the absence of hippocampus bilaterally.
Yeah. I think I remember that example from my school days. So this person would, after the procedure, they would meet someone new and then, meet them again the next day, and they wouldn't remember that they met them the previous day.
That's exactly right. So, but that's with the whole hippocampus, or let's say major parts of the hippocampus. What's interesting is oftentimes there have been reports of individuals with unilateral damage. They can compensate with a single side, which is an interesting. I don't know whether or not the clinicians have dug in deeper to that, where they can actually tease out some differences between them or not, but it's much less with a unilateral damage compared to a bilateral damage.
And you mentioned earlier, I think that you, in experimental animals like rodents, you can just inhibit neurogenesis in general. When you do that, what kinds of deficits do you see?
Yeah, this is where I was getting into this discussion of pattern separation, where they can make distinctions between. So one of the classics is a fear conditioning test, where the animals are given a slight shock in a chamber with certain features about it, has very distinctive features. And if you put the animal back into that after 24 hours, they will freeze, anticipating they're going to get shocked, right? A normal animal won't freeze because when they see it the first time, they're wandering around, sniffing, smelling. But if they're put back in 24 hours to that same environment, they freeze. And if you eliminate neurogenesis, you put them back in the box 24 hours later, and they walk around the box as if they've never seen it before. So they don't see this environment as something that they have previously seen. So but interestingly, you can put them into a chamber that is slightly like this one, but different. And a healthy animal will still freeze a bit because they're seeing the similarity between it.
Mm-hmm.
But an animal without neurogenesis doesn't recognize it even further without. And then if you put them into a chamber that is completely different, even the animal without neurogenesis can figure that out. So it's really only looking at those events and locations and emotions that's making the distinction
slightly different.
Slightly different. Yeah, that's the sort of the nuances of life.
Yeah. Yeah. So again, I mean, it's this idea of pattern separation being connected to generalization. Your ability to make fine scale distinctions also impacts your ability to generalize and determine when things are part of the same category or context and when they're not.
Absolutely. That's exactly the way I see it.
So, based on everything you've told us, everything everyone's always getting older, obviously. I'm sort of, I guess, early middle age. As I go through the rest of my life, what are the major elements of a person's lifestyle that they can easily control that will help preserve neurogenesis and maintain it at healthy levels throughout the second half of their life?
4 things. Get plenty of healthy exercise. Expose yourself to novel experiences, learn new things, travel, just learn things, acquire new information. Clearly have a healthy diet. And by that, this means, all the things that people are talking about in terms of processed food, because we know the glucose, glycogen, metabolic activities are key to this whole process. So metabolics is big, and you can impact metabolics by diet, by not dieting necessarily, but by eating appropriate foods. And the first is to live in a social and low stress. Try to keep your anxiety levels down. Try to keep your stress levels down. Avoid places where are going to make you tremendously stressful. On the other hand, a good amount of excitement and challenges are important, but not to the point where it's causing the release of corticosterone and other cortisol levels rise in your blood. You want to keep away from those kinds of things.
I would imagine. I almost see an analogy with you said intermittent fasting can help on the diet side. It's almost like novelty in some sense is almost like intermittent as opposed to chronic stress. Yeah.
There's good stress and there's bad stress. Stress is a continuum too. So that's what I was trying to say. Exactly right. Is it this low level, excitatory activity, novelties, learning a new thing, the frustration of, learning a new language, and the early stages are tough, but staying with it and acquiring it, it builds up. And then there's sort of growing evidence that socialization is a key feature as well. So, social isolation has been evidenced to show decrease in neurogenesis as well. So highly social, low stress environments are part of it. So that may be 5 if I separate out sort of anxiety from the social environment, but I sometimes link those sort of things together.
Interesting. Well, Professor Rusty Gage, this is fascinating stuff. Thank you for taking the time. Is there anything else you want to say or maybe reiterate based on anything that we discussed today?
Trying to think. I think not. I think that was good. I hope it was understandable, and recognize that many of the things that I said were my opinions or my interpretations of the data, and there's lots more to discover. So it's a great area for young people to get into, because it's thought provoking, and yet there's really good underlying molecular mechanisms and circuitry questions. So for neuroscientists, it's a rich area in an area of importance for cognitive decline as well.
All right, Professor Rusty Gage, thank you for your time.
Great, thank you very much. Have a great day.
This episode episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it with the first meal of each day, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure. And vitamin D is super important for the proper function of the immune system and for a variety of other things. And there's even evidence indicating that vitamin D deficiency is correlated with more severe cases of COVID-19 in those who get infected. Every time I go into the doctor each year for a checkup, I'm always told that vitamin D deficiency is very common and I should be supplementing on a daily basis. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.